estimation of time since they shared a common ancestor.
So it's a pretty old
Hippos? No, it's just that in terms of, it's a question of when they parted ways. The hypothesis is they once shared a common ancestor, i.e. they were the same species. We now have modern whales, and we have modern hippos. But it's been a long time since they parted ways. Whereas, if we wanted to put more resolution onto this, we could start saying, okay, here's the toothed whales, modern toothed whales, okay? And then we might also say, okay, here's dolphins. And then as we zoom in, we get, it's a prediction or a hypothesis of when organisms that we see last shared a common ancestor. Does that make sense? Okay. Other questions? Good question. Alrighty. How you doing? Okay. So, original hypothesis still on the board, Darwin's original hypothesis. The prediction is that speciation occurs between closely related groups of organisms. Did I leave these slides in? I think I haven't made a mistake here. We'll see in a sec. So the question is, okay, can we look for evidence of speciation in Project Gress now? Okay. The result is that, yes, we can actually observe speciation in many cases. And one noticeable example is this idea of what is called a ring species. So species that are distributed around a ring of suitable habitat. I'm hoping I left in the slides. I may not have.
It's not in, we're just different.
They're not in yours? Okay, I'll make sure that I put up another copy for you to look at. So just feel free to relax and listen. What ring species are closely related groups of species. So in this case, every colored bar is a species. Neighboring species can interbreed with each other. So these ones can interbreed. So they kind of confound the definition of a species, right? Because they're actually interbreeding with each other. These ones interbreed with each other just fine. These ones interbreed with each other just fine. But the two species on the ends, should you put them together with one another, will not interbreed. They are true species in terms of at the end. But every intermediate step along the way successfully interbreeds. Well, all it would take, hypothetically, is some sort of event to sever those intervening populations, and you, voila, have two good species that are no longer interbreeding with each other. You can actually see examples in nature of this thing going on, such that here's a ring species circling some unsuitable habitat, say a mountain range or something like that, where even where there is overlap between the two species at the end, they still cannot successfully interbreed with each other. Okay. More to the point, and stronger evidence still, is something called phylogenomics. Phylogenomics is basically using DNA sequences excuse me, to test hypotheses of relatedness. So, to go back to this example, why are whales and hippos now proposed to have once shared a common ancestor? That's pretty hard to tease out just by comparative anatomy and such, right? They're very different organisms. But nowadays, we sequence DNA from all sorts of things. And this hypothesis was actually formed based on comparison of DNA sequences between the two organisms. And we're getting better and better at this in that we can now begin to compare whole genomes. So the complete DNA sequence of an entire organism's code, one with the other. And we can line them up and say, are they different? Are they not different? It's kind of like comparative anatomy, but at the nucleotide level. Just how similar are they? How different are they? Okay. So, DNA sequence of organisms thought to be closely related to each other based on anatomical or morphological criteria. You can test those hypotheses by lining up their DNA sequence one with the other. Seeing ones that are predicted to be more closely related, i.e. more recently were the same species, should be genetically more similar to each other than organisms that have been separated for a longer time. Okay, so the test in this case is to sequence entire genomes and just compare sequences one with the other. Is everybody okay with the process here? What's going on? Feel free to flag me down. I know it's kind of a whirlwind tour. If you've had any religious studies courses and you've studied textual criticism, Does that ring a bell for anybody? Textual criticism is basically looking at different copies of manuscripts, say of the Gospel of John. None of the manuscripts that we have are absolutely identical one to the other. However, they're very, very similar, and errors can get copied by scribes. So you can begin to group documents together and say, this one looks like it's a copy of this document because we see the same errors, as opposed to hypothesizing that those errors arose independently between two different scribes, that kind of thing. Molecular phylogenomics is essentially textual criticism on DNA sequences. It's the exact same process. If you think of DNA code as a text, it has an order, a syntax. You can compare one with the other. Okay, so what do we get when we start sequencing organisms? The reason why we've been doing so much of this lately is because we have all the leftover infrastructure from the Human Genome Project. We have all these DNA sequencing machines that aren't sequencing human genomes anymore. So what we can do is we can just run organism of the day through the sequencer, as it were. So we're starting to sequence even more and more obscure things. So what sort of result do you get from that? Well, the overall predictions of relatedness based on other criteria are supported by this approach of looking at DNA sequences. Now, to be truthful, at really high levels of resolution, you're looking at very closely related groups of organisms, molecular sequencing has shuffled a few things around because it has a greater resolution power than do other criteria. But on the whole, for larger, more separate groups of organisms, it's quite easy to distinguish, and phylogenies hold, or the degrees of relatedness hold. Okay, so here's an example from two organisms that are thought to be closely related based on morphological criteria. Okay? This was noted in the 1980s. This is simply taking chromosomes and staining them with a chromosomal dye. So you can look at the morphology of the chromosomes and see how they compare one to another. So in this, what we've got here is in each grouping, we've got chromosome from one organism here, and then a chromosome from the other organism lined up with it. You'll notice that there's quite a striking correspondence. It's not perfect. There's some areas that seem to have been inverted, flipped around. That can happen to DNA sequences. But there's other regions that seem to be very, very similar one to the other. There's one noticeable difference, though. This one here. What's present as two chromosomes in the one species is one single chromosome in the other species. So that immediately suggests some hypotheses. What sort of hypothesis do you think it suggests? Well, it suggests that a chromosome fusion event happened at some point between these related groups of species. Okay? It suggests that this is the ancestral form, these two separate chromosomes, which in this other related species is fused together into one chromosome. Now, I deliberately blacked out the species in question, and the reason being is that this example hits close to home. This is a comparison of chimpanzee chromosomes with human chromosomes. Okay, so it's testing the hypothesis that chimpanzees and humans at one point shared a common ancestor. That's the test. That has been long predicted based on comparative morphology, anatomy, and physiology. The DNA sequencing now allows us to make this test at the molecular sequence level. As disturbing as this might be, theologically to some, it doesn't change the facts that we're dealing with. Okay, so that previous paper was 1980. This paper is 2005. Yeah, question, sorry.
So when they're doing this,
yeah.
are they just looking at the chromosomes, or are they looking at the whole DNA?
They're looking at both. The question was, are they looking just at chromosomes or the whole DNA pattern? DNA in our cells comes in linear sections, and each chromosome is one linear piece of DNA. So that original study, what they were looking at is they were simply taking whole chromosomes and staining them with dyes that stick to DNA, and then just looking at the pattern that they see. Some regions stain more heavily, some regions stain more lightly, so you end up with this banding kind of pattern. So it's just a rough look to say, okay, how similar do they look one to another just based at like a light microscopy level. So they're not sequencing anything, they're just looking at the physical shape of the chromosomes. Does that answer your question?
They use sequencing?
Well, they couldn't in 1980, but we did it in 2005. So in 2005, the